Our next speaker is Professor Ioan Andricioaei. He graduated from Boston University in 1999, and he's now a professor in the Chemistry School of Physical Science in University of California, Iowa. And his major research interest is developing sampling methods in trajectory space, and also applying those methods in, for example, simulations of DNA binding machines. So today, the topic will be the interplay between local and global DNA dynamics using computer simulations.
Hello, everyone. Thank you very much to the organizers and to Lige in particular for the invitation and the opportunity to be here. I am very excited to be in Shenzhen. And the reason I'm excited is because of the feel of growth that I sense through the larger Shenzhen area as a city, but also at the Chinese University of Hong Kong, Shenzhen, and in particular at the Warshall Institute. The reason I am excited also has to do with the fact that I'm coming from an institute and from a university that has experienced a similar growth over the past 50 years. I'm coming from the University of California, Irvine, which did not exist some 50 years ago, and it was developed to satisfy the needs for education of science of a growing California population. And it is this excitement to be part of something that you're contributing to build that will endure that I feel happy to be here and share with you some of my research in the area of molecular biophysics via computational means. Irvine is a little, is relatively close to here. You just have to go over this body of ocean for about 12 hours in a plane, but otherwise it's relatively close, and this is the overall layout of the campus. Somewhere around here, I understand your dean has spent about a year some time ago. So it's a place that is currently growing, and this excitement towards development is reflected in the science there. So what I'm going to talk to you about today is the way in which we simulate the importance of motions in DNA, motions that are important both at the level of local base pairing, right? We have A pairs with T and C pairs with G, and we have a lot of important dogmatic molecular biology. DNA makes proteins, and DNA makes RNA, and RNA makes proteins, so DNA is the essential genetic carrier for life. But away from that local level, you have global conformational changes that extend over micrometers in scale, and microseconds and milliseconds and even seconds in duration, that are also important to biology. For example, DNA is wrapped around chromosomes in situations that look more like these nautical knots rather than the straightforward picture of a double helix. Gene expression is produced by looping such large global conformations. We have DNA that's supercoiled, linked, or in catenanes, and the DNA is unzipped in replication or is squeezed very tightly with pressures that exceed 10 times the pressure in a champagne bottle inside the capsid of viruses. So all these things are important, and they are on one hand exciting, on the other hand are quite challenging, and point to the need to do multi-scale simulations, right? You can treat with atomic detail on supercomputers the local motions, but you can access them only up to microseconds for, say, a nanometer length scale. But longer than that, you have to go to coarse-grained levels, and you have to switch your tools to other tools for pen and paper, and very often we also need this tool, to build up cross-scale simulations and treatments of the dynamics that address the dynamics at large scales. In terms of fundamental descriptions of in our group, we focus on path sampling, on understanding what the kinetics of conformational transitions between these important conformations, not only of DNA, but also we apply it to proteins and nanoparticles, but this talk will focus only on the DNA. So we study the contribution of trajectories, these objects, x of t, where x is the 3n-dimensional configuration of a biomolecule, right? And this is the field in which Ligier works. We're trying to understand what are the fundamental objects, these time correlation functions, from sampling trajectories x and t on this pedagogical description of the rugged energy landscape, right? So X represents one of the highly dimensional configurational space, right? 3 times the number of atoms, where the number of atoms is of the order of 10 to the 5. And here we have the potential energy, right? And the typical cut through this hypersurface is very rugged. We have a large distribution of barriers that are much larger but also much lower than KT, and it is very often that the simulation gets trapped in these minima, and we need tools such as the ones that Professor Hansmann has described to be able to escape these barriers and figure out what is, in the realm of thermodynamics, the average of interest, thermodynamic average. What is the thermodynamic average? You can change the rules of the game, as Professor Hansmann is doing. You can run at a higher temperature, or you can run on an effective potential. And you're sampling something that is computationally expedient, but is artificial in the sense that the weight of the points x that are sampled via enhanced sampling is wrong. Wrong, but you know how wrong it is. This is the wrong distribution, and this is the correct distribution. So what you're going to do to get the exact average is to divide by the wrong weight and multiply by the correct weight. You are reweighting, you are correcting, so in the end, you get an exact thermodynamic average. This is exactly what we're doing in the realm of kinetics. We're generating, via some means that I won't go into, we're generating some fast trajectories that sample another system that's artificial, but computationally expedient, it's fast. Then these trajectories, which now are not points, but are spaghetti lines, if you want. They are space-time objects, right? They are these trajectories that connect places on the potential energy. And they are wrong in their way, meaning the probability with which we generate them is wrong, but we know how wrong it is. It is wrong by the ratio of the probability with which we generated that path, and we know what that exact path would have as a weight if it was generated on the real potential, not on the artificial one. So we're going to multiply by it. And in a similar way, we're going to get the time correlation function exactly for something that takes, say, milliseconds just by doing nanoseconds, by dilating time in this manner. Okay? So this is the only theoretical slide that I'm going to show you. Now I will walk you through a zoo of systems on which we applied our computational techniques. And I'll start with not simulation, but actually with experiment. Very excitingly, the field of single molecule biophysics is advancing by heaps and bounds, mainly because they are able to do a very fundamental thing. Experiments are able to discern for the first time not just averages, but distributions of properties, right? So it's as if instead of measuring what is the average height of the students of Chinese University, Hong Kong, you can actually go and plot what the distribution of all the heights is, and you can find perhaps subpopulations, and you can track how this subpopulation of height changes as the season changes, as vacation comes, and so on and so forth. This is fundamentally what single molecule experiments are doing, and ironically, we as computer simulation people have been doing this from the beginning, not because we want to, but because we're forced, right? We cannot work with Avogadro's numbers of molecules. So it is a very basic and a natural relationship to look at single molecule experiments with the tools of molecular simulations. And this is what we're doing here. We're analyzing so-called single molecule pooling experiments. In this case, we have a folded RNA structure, which is small here and you cannot see, but the experiments cannot see it either, right? So they pull on it with 2 beads. This is the scale, right? So the beads are very large compared to the molecule that you're unzipping. Yes, and this is actually the structure of the RNA. This RNA has some. This is coming out of the middleman position, is doing some catalysis. It's a ribozyme, actually. And experiments are able to track the force between the beads as a function of the extension between the beads. Okay, so these are typical nanometer extensions, and forces are on the piconewton scale. Okay. Now, the problem with the experiments is that they are done at force conditions that are relatively large. Regardless of that, when you want to understand why are you doing this, why are you unzipping an RNA, because you're not interested in unzipping, you're actually interested in folding of RNA. So you're unfolding it and hope to understand something about the reverse process. The trouble is that the force is much larger in reality. Of course, in vivo circumstances, you don't have any force. But then also low forces are not well sampled. When you pull on something, you don't know if you're pulling with large forces, you don't know if you're pulling on these tethers, on these handles that are also DNA molecules, or if you're pulling on the molecule itself. So you need some theoretical way to tell you what the kinetics of this unzipping is, of this unfolding. What is the kinetics of this if you only have high force conditions? Can you tell me what the kinetics would be under lower forces or perhaps no force at all? This is the idea of the project. So the way we do this is to, I'm not going to talk about theoretical details. We take each experiment at a time. These are single molecule experiments. Each line that you see here, each spaghetti noodle, is a repetition of the unfolding under other conditions. So you pull and unzip, and you let it relax, and you pull and unzip again. And every time you do this, you measure a different individual, right? So you're getting a different trajectory. That's what's exciting about single molecule experiments. And so let's zoom in on an example here. Let's say that you pulled this molecule with 530 nanometers per second. That's high pulling. But you would like to, and you also pull it at a lower force condition, right? And so from the high force condition, as a test, as a positive control, let's say, you want to figure out the kinetics at 360 nanometers per second from data only at 530. Of course, you know the exact kinetics at the lower force condition. This is the green curve for the autocorrelation function of some unfolding measure. Yes, so you know the green curve. You also know the red curve. You would like to get the green curve only from red trajectories, from red experiments. And the way we do this is, if you remember the theoretical slide, each of these trajectories, it has a weight in the experiment. So we calculate the weight of that trajectory, and we pretend it's part of the green ensemble, where that red trajectory would have another weight. So we're going to divide by the red weight and multiply by the green weight, and in the end of the day, we're going to get the red time correlation function that's actually in the green ensemble. We're going to extrapolate high force experiments to calculate for us low force time correlation functions, which are the fundamental object of kinetics. Now I'm going to switch topics and tell you about local DNA dynamics. We have looked at very interesting motions that take place at the level of the base pair. Base pair, which was the structure of which in the DNA was famously identified by Watson and Crick in '53. This is Watson, the biologist. This is Crick, the physicist. He has a slide ruler here with which he happily measures the distances and static structures of the DNA. DNA has proven to be very different from that. Another scientist, albeit several hundred years earlier than that, realized that things are moving. Things are still moving, as he famously scribbled. He also has something in his hand. He has a nail this time, a nail with which he scribbled the famous words, "It is still moving," on the walls of his own cell. So with more dire consequences, he realized that things are moving. So things are moving. Actually, we find that they're moving site-specifically. Okay? Things are moving in the DNA world differently if an A is paired than a T, as in the case of C pairing with a G. Okay, so things are moving, and the fact that things are moving also has been recognized in the Nobel Prize Award in 2013, given to scientists, including the founder of this institute. Okay? So, what does molecular dynamics have to do with DNA? We looked at this motion in the context of the base pair. So here I introduced a little bit of molecular biology, although I'm sure I don't need to very deeply. So I'm just going to say that air pairs with T and forms 2 hydrogen bonds, and C pairs with G making 3 hydrogen bonds. And one is an acceptor and the other is a donor. But if you look at the molecules, there are other acceptors and donors that could in principle form hydrogen bonds, right? So only certain combinations of hydrogen bonds are possible. But in this pas de deux, we see that if you have various ways to form bonds, right? You have an acceptor and a donor, but note here that the partner's right hand is coupled to the right hand of the ballerina, while here the right hand is coupled to the left hand and the right hand is coupled to and the feet are also exchanging interactions. So you can form hydrogen bonds in CG in a variety of ways. You just need a flip over of one of the partners. This is the essence of a Hooke's base pairing, right? So for example, for AT, you can hydrogen bond this way, or you can hydrogen bond this other way. For CG, similarly, you can hydrogen bond with these 3 base pairs, but the cytosine can flip over around the glycosidic angle and form other hydrogen bonds, right? So these are called Hooke's base pairs, and they've been known for quite a while. And we were part of a collaboration that showed that this conformational transition actually occurs in naked DNA. So for a very long time, people thought that, yes, these Hooke's conformations do occur, but they occur only in very strange situations when a protein binds to a DNA and distorts it in such a way that the flip over is induced. Yet, using NMR experiments, relaxation dispersion experiments in particular, that are able to probe microsecond to millisecond motions in very weak populations, my collaborator, Hashim Al-Hashimi from the University of Michigan at that time, now at Duke University, has shown that these Hookestein conformations do exist in naked DNA, that is in DNA free of any protein, just that they occur only 1 in 100 in population, and the time to flip over is microsecond to millisecond. Okay? And what we have done to verify this was to calculate a two-dimensional free energy surface, a two-dimensional free energy surface that has as parameters the glycosidic angle. Think of this as the flip over angle, yes? And the theta angle is the flip out angle. The virtual dihedral that tells you if the DNA flips out or not. The situation is such that if I have an AT base pair, right, and this is A, I want to induce a conformational transition in which it binds like this. But there isn't sufficient space inside the duplex because there is a base pair up and there's a base pair down, so it cannot flip inside. What it needs to do, it has to get out and then flip over and then flip back in, but it can flip out through the major groove and it can flip out through the minor groove. Moreover, it can flip over clockwise and it can flip over counterclockwise. So this should be reflected on the free energy surface, and we found that this free energy surface has minima, as you expect, in the Watson-Crick. There's a higher minimum in the Hookestein, and the relative population, if you calculate the Boltzmann weight of this state relative to the Boltzmann weight of this state, it is about 1 in 100. But then on this two-dimensional surface, you also have paths, the kind of paths that I'm working with Ligia on to try to understand in more detail. These are paths to go from Watson-Crick to Hookestein to flip over, and there are 4 of them, right? Major groove, minor groove, 2 times 2, which means flip around positively and negatively. So we identify these paths, and we are putting them in connection to the NMR work. Okay, so how much time do I have? Another 15 minutes? Okay. Now we look at something different. We want to understand this propensity to form Hookestein base pairs in a global context. You know, if you have a straight DNA, then you can imagine that the A in the AT pair will flip over with some propensity. And if you have a nucleosome or you have tightly bent DNA, then the propensity is different. And that is what you can look, and you can imagine that the entropic effect, the entropic changes of the local conformations of DNA are going to be important, as well as the relative positioning of the DNA in the bent form. Because think about it this way, think about walking along the helical axis. As you walk along the helical axis, you're also twisting your reference frame in a circle. So you're going to have the major groove outside, then you're walking over, and then you're flipping, then the major groove will be inside, then it'll be outside again. You're doing this barrel roll, and whether you are in the major groove and or your minor groove, the major groove is going to be opened even further if it is outside, and is going to be squeezed in as you're walking around this helical axis in a circle. Okay, so these are effects that can be addressed with molecular dynamics at the atomic level. So I walked you through 2 animals in the zoo, right? I showed you how we analyze single molecule experiments on RNA and DNA pooling, and also how we understand local DNA dynamics. Now we're looking at a totally different system. This system has to do with DNA presented with dendrimers. Dendrimers are man-made polymers, and in this case, We look at PAMAM dendrimers, which are very positively charged on their outside. They look like spheres of growing radius, right? You have generation 2, generation 3, generation 4 dendrimers, larger and larger. And they have terminations that are positively charged under normal pH conditions. So for example, one dendrimer that we looked at has 32 positive charges, right? DNA, on the other hand, has 24 negatively charges for a 12-mer. Yes, for the respective size of DNA, respective to the dendrimer. The motivation was to work on this topic with my former graduate student Mariah Mills, who is now at the National Institutes of Health, moving on to her independent faculty position at the University of Missouri. Motivation was to understand again single molecule experiments, this time of another sort. In these single molecule experiments, what is produced is a condensation of DNA. DNA in the absence of these dendrimers, right? Dendrimers are highly positively charged. 32 plus DNA is chock full of minuses, -1 on each phosphate. When you present such a polymer with dendrimers, the dendrimers are going to bind and compact DNA to a volume that in the most compact state is 1,000 times smaller hydrodynamic volume than the hydrodynamic volume of the starting elongated DNA. And this compacted DNA form has now the potential to be pooled with single molecule experiments of the type done, for example, shown here in Carlos Bustamante's group at UC Berkeley. What they do is to attach again beads at the end of the DNA, and the beads are optical traps, so they can be moved up and one can measure the tension. And they take the compact form of the DNA when the dendrimers are bound to it, and they pull it taut until the dendrimers pop out, and you have extended DNA, for which again you measure force extension curves, right? So here is the compact form of the DNA. You pull on it, right? The DNA starts to extend, extension increases, the dendrimers pop out, and then here you fully extended DNA, and no matter how much force you continue to apply, the extension remains constant. What's interesting, though, is when you come back, the DNA refolds, recompacts, but goes on a different path. This is called hysteresis, and it's a signature of non-equilibrium. This is the same kind of effect that you feel macroscopically when you unzip Velcro tape. Say you have shoes that are tied with Velcro tape. If you pull on the tape quicker, you'll need a larger force than if you pull on the tape slower. This effect is basically what's happening here. And the area of the hysteresis is related to the dissipative heat that you produce in such cases. Okay? Now, try to simulate that if you can at the atomic level, because we're talking here about very long DNA molecules, right, that exceed the capabilities of even the fastest computers. So what we have done is to perform a combination of atomistic simulations with some sort of mesoscopic continuum model. And the simulation proceeds as follows. This is a dendrimer, right, and it binds to the DNA. So what we will do is to simulate the local dynamics and energetics of the dendrimer DNA system. We're going to calculate the free energy as a function of the distance between one dendrimer and one piece of DNA only. We're going to do this in various orientations and whatnot, but we're going to get free energy profiles. You see a minimum here. This is the most favorable state. And this is the barrier needed to dissociate the dendrimer from the DNA. These are obtained via atomic molecular dynamics. It has two parameters. It has a height of the free energy, and it has an extension. Then this free energy, which is also called, by the way, a potential of mean force, is called a potential mean force because it is the potential whose gradient is the mean force. In other words, if we take the derivative of this curve with respect to the extension, we're going to get the mean force. The mean force, which, by the way, is also measured in the experiment. So these are the piconewtons that can be directly compared with the piconewtons on the force extension curve of the experiment. Right, so now we also have forces from the simulations. And we introduce this mesoscopic model where we have bound dendrimer and unbound dendrimer going across a barrier, but the height of that barrier is tilted by the applied force. This is using the so-called bell curve model, right? So if I apply a force to this potential, it's going to tilt. The higher the force, the more tilted the potential. So now we can combine the atomistic simulation with a Monte Carlo propagation model of a master equation in which we only have two states, bound and unbound, and the kinetic parameters of this Markov state model come from, without fitting, the atomic simulations, the barriers that gives us the rate and the distances that modulates the speed with which, with the speed with which DNA advances. These parameters can be put together, and we can create the force extension curve that we can directly compare with the experiment, as you see here. What is remarkable is that the fit to the experiment is good without an actual fit in the microscopic parameters. We did not fit the parameters of the microscopic interaction. With the parameters coming from the microscopic interaction, we were able to get good agreement with the simulation. Okay, so now the last topic has to do with another type of multi-scale representation of the DNA. In this case, DNA is represented as an elastic rod with the so-called Kirchhoff theory of elasticity, right? So it would be great if we could do hundreds of thousands of base pairs with atomic simulations, but we cannot. We can only do a little bit of this entire piece with atomic simulations. So let's do that little bit of simulations. Let's take a piece, cut a reasonably small piece of DNA, or large, depending how you want to see it, one that you can actually simulate, and calculate from this atomistic simulation continuum parameters that will work with Kirchhoff's elasticity theory. So now we look at the DNA as a rubber rod, as an elastic rod. Okay, this elastic rod will have a certain set of parameters that describe its bending. How much energy do I need to put in to bend DNA, and how much energy do I need to twist DNA? And with these parameters, these are this is a 3x3 matrix that describe this stiffness of the DNA, these bending and twisting parameters. We create recreate what's in effect some sort of elastic continuum, right? If you have a spring, the force constant times the square of the fluctuation gives you the energy. This is it, basically. This is the force constant, B is the force constant, and this is x, and x, this is x squared. But x here is actually the tangent to the helical axis. Okay? When you integrate this along the contour of the DNA, you get an energy, right? And now that you have an energy, you can do the same things that you do with the energy in molecular dynamics. What do you do with energy? You take its derivative, you have the force, and you write ma equals the force, meaning you write the balance of the linear momentum. And so these are the Newton equations, if you want, of motion of the rod. You have balance of the linear momentum, but you have also balance of the angular momentum. And this gives you dynamics of the elastic rod. So with these dynamics, we can look at larger pieces of DNA than were actually simulated. And it is interesting, for example, to look what happens with the DNA that's packed inside capsids of viruses. Okay, here you have a proteinaceous icosahedral capsid in which you put in DNA, and DNA is highly compacted here. As I said, this is about 10 times the pressure in a champagne bottle, right? So if you'd open this cork here, DNA would go out with a similar force at the, of course, compare the microscopic level. So in a cryo-EM picture of the bacteriophage Phe29 filled with DNA, it was seen that there was a very intriguing twist. There was a very intriguing structure in an opening of the pore through which DNA is ejected. And the question from the experimental group of Tim Baker and collaborators at UC San Diego was, what is in there? What sort of structure is in there? And the hypothesis is that it is DNA. As DNA goes out, it kinks and bends in there, kind of looking like this. But if you think about it, this is a size, this is mini-circle DNA that's smaller than the smallest mini-circle exists, right? For a typical mini-circle, the smallest mini-circle that you can bend in free conditions, right, without any protein, is about of the order of hundreds of base pairs. Here you have about 20 to 30 base pairs, so much shorter perimeter. Okay, what sort of structure is in there? Of course, this is made possible by the presence of the protein around it that keeps it such highly bent. So what we did was to take the elastic rod model that we squeezed inside the confines of the cryo-EM density, used as boundary conditions to preserve the DNA structure. So we started from the top, yes, and we pushed this elastic rod, pushed and pushed more and more until it started to kink and bend and fill in the hourglass figure, and produced a variety of DNA ensembles. And for each of these members of the ensemble, you can build then the atoms back. You can let the atoms grow back in the elastic continuum, because the elastic continuum has a helical axis, and then you just put bases perpendicular to the helical axis, build, let the atoms grow around, and do minimization and molecular dynamics equilibration, and figure out what the atoms should look like. And then from the atoms, you can get a step further, and you can back calculate what would the cryo-EM structure look for that particular member of the ensemble. This is how it would look. And then you can average, you can do a linear combination, a weighted combination of these structures, and you can compare the resulted average from the ensemble with the actual cryo-EM data and come up with explanations, what sort of members are there in the ensemble and what are their energies, okay? And also you can do dynamics. This is the last slide that I'm going to show. This is the last topic that I'm going to show. You can actually illustrate how when you have supercoiled DNA and you cut that DNA, say by topoisomerases and such, you can produce molecular simulations of the supercoiled relaxation of the DNA on time scales of microseconds to milliseconds that actually take in the simulation as much time as you needed to watch this movie. So this is sort of real-time simulation. It's done at the elastic continuum level, just that the parameters were calculated from very extensive molecular dynamic simulations on pieces of DNA. Okay, so this is what I have showed to you so far. I want to acknowledge the work of various people who have contributed to these topics. In particular, Mariah Mills' work on the dendrimers, and Marina Taranova did the simulations of of DNA minicircles together with the group of Noel Perkins at the University of Michigan. We developed the elastic rod model. And with Hashim al-Hashimi, we are continuing our collaboration on the role of local DNA dynamics as a second tier of genetic information. So I stop here. I thank you again for the opportunity to be here and also for your attention.
It's a wonderful talk. So any questions? Yes, please.
Very interesting talk. So I'm wondering, what are the strategies to make the CUHKSZ pair more stable than the Watson-Crick pair, if there are?
What makes it more stable?
Yes, if it's possible to manipulate.
Yes, very good question. So in the general case, the CUHKSZ is less stable than the Watson-Crick by a ratio of 1 to 100 in population. However, when proteins bind, like P53 is an example, it distorts the conformation of the DNA in such a way that the flip over that the CUHKSZ base pair is more stable. But that happens in the presence of the protein. If you take the protein away, then it reverts to the Watson-Crick. So this is a topic that we're currently trying to understand. Is it just the fact that the DNA is bent that makes the CUHKSZ more favorable? Or is it the contribution, steric and charge-like, of the protein that also induces this conformation? Okay? What I should also say is that we are now scanning the entire protein data bank for cases in which crystallographers have assigned a certain orientation for a base pair, thinking that it's Watson-Crick, although the data was very noisy. In several examples, we can find evidence that actually, should they have modeled the CUHKSZ conformation, that would have fit the data better. Okay?
Well, I can actually add to that. There are those pseudonuts actually stabilized by CUHKSZ interactions, and they can drive ribosome frame shifting. So it's proven by also your single molecule technique that it's quite strong.
Yes, yes, and in the context of RNA, this is another thing we're looking at. I've talked about DNA CUHKSZ conformations. In naked RNA, CUHKSZ base pairing is not even seen in the experiment, meaning that should it exist, it's a population much weaker than 1 in 100. But RNA can also fold in more mysterious and complicated ways than DNA, and so there, you know, these CUHKSZ base pairs arise naturally.
I do have a couple questions on your other parts of your talk. On the single molecule optic tweezer pooling experiment,
yes.
Although you published 12 years ago, I still find quite new and interesting. So the question is the goal and also another methodology. So your goal to do that is to speed up the pooling because you need to sample multiple times, and then to project slower pooling. Was that the goal, or what is the goal doing that?
The goal was the following. So you are talking about the extrapolation of a single molecule unzipping
in different
from
forces.
High force to low force.
Low forces.
When experiments pool with high force, you don't know, so the RNA is connected to the beads via DNA linkers. And so those linkers are fairly long, and they are longer than their persistence length. So they're not entirely pulled taut. They are wavy. When you're pulling with high forces, of course, you don't know what you're pulling on. Are you pulling to extend the linkers, or are you pulling to unfold the RNA? Yeah, so you get some results from the high force data, and you think it's, or you interpret it, the experiments interpret it as saying something about the RNA unfolding. But it could be a contribution from the DNA extension or straightening, right? As they pulling on the entropic spring. So we would like to be able to extrapolate those from high forces to lower forces. Forces that are not accessible in the experiment.
The real force on the shooter you're pulling apart. So
Yes, is not so it is for
extract the real force.
Yes.
Okay.
Yes.
Okay. So the way you do it would be using so you get the ensemble of the high force, and you use the simulation to get the weighting for the low force and high force.
Yes,
then
so
back calculate the low
the
force.
trajectories are generated, if you want more technical explanation, using the Langevin dynamics. And in the Langevin dynamics, you have an external force there. Of course, you have friction and dissipation. But this external force produces a Langevin trajectory with a certain weight. If you had another force, it would produce another weight to the same trajectory. Right? So this is what we're reweighting. We're interested in the ensemble of Langevin trajectories at low force.
Thank you.
Hi, thank you very much for the stunning talk, Professor. So I have, thank you. So I have a question about the trick of elastic role model of DNA simulation like that. I think it's a quite good idea to simulate DNA with such a I think it's kind of a global winning model. Is that a coarse-grained model for simulations? The first question, sorry.
You can call it coarse graining if you want.
Yeah.
Coarse graining is so this, right?
Yeah. Yeah.
Where we are actually better shown here. Yes?
Yeah.
Is this coarse graining? So in the traditional canonical definition of coarse graining, you would, if these are atoms, right, you would say, okay, let me take 10 of these atoms and replace them by their center of mass. So now I have 10 times less atoms, and those center of masses interact via some sort of integrated over force, or some sort of a renormalized force, right? You recalculate a potential of mean force. Right, and here what we have is we don't have centers of
don't
mass.
have centers.
We have a continuum described by this. This is actually a functional. Yes, it depends on this function, which is a vectorial function, where S is the parameter along the axis, right? So I have a function in space, I have a line, and that line has a potential.
So
there's an energy. This is the energy ascribed to that path, to that line. So that line, as it's moving, it has going to have different energies. Yes. So if you want to call this coarse graining to the extremum, right? We discretize so much that it becomes continuous.
So your parameters will all depend on the sequence of the DNA, or otherwise
in principle, they can. Here, they don't. Here, this parameter, right? This is a set of it's a 3x3 matrix, 6 independent elements. They do not depend on sequence. You can make it if you want, but this work does not make them sequence dependent.
Okay, great, thank you.
You're welcome. Yes.
Thank you, Professor. Very interesting talk. I was a little interested about this elastic rod model. I was wondering if you change the shape of the constraint space. Hello? Okay, change the size of the restrained area. For example, if it doesn't like a rod, it's like a sphere or like a cubic. Will be the DNA constrained into other kinds of the certain shape? The second question is about, I was wondering the perception about this model. For example, if I want to use this rod model into the crowding effect,
crowding
effect, is that still workable? Yeah.
Okay, so regarding the first question, which is whether different spatial constraints produce different DNA structures, the answer is yes, absolutely. For example, here it was exactly the shape of the constraint. The shape of the constraint is the boundary of this empty hourglass figure. That produced the shapes here. If in other instances I don't have these constraints, but I have, say, for example, a situation in which I just take a piece of DNA, right, and my constraints are only here and here, and I'm going to do this, right? I'm going to rotate clockwise in one direction and also clockwise in the other boundary, then I'm going to produce other structures which are called supercoils. Okay? So that's another example. If I have constraints that say in nucleosomes, if I have spherical constraints and I wrap DNA around it, that will produce, cylindrical structures. Regarding your second question, which was
I was just wondering if this model can use in the other area, for example, the crowding effect.
Crowding effects. So if crowding here can be modeled simplistically by placing objects. So if you want to understand supercoiling in a crowded effect, you can just put here balls of hard spheres or something that distort the conformations. You can also attach, if you know that in a certain place on the DNA sequence a protein binds. In this case, if you know that topoisomerase binds on the supercoil and cuts one strand, and that cut strand rotates and releases the supercoil, that you can also simulate. In fact, this is what's modeled here. The change of the constraint is applied in the simulation. The constraint is actually coming from a protein. It's not exactly for crowding, but this kind of applying boundary constraints, even time-dependent or position-dependent boundary constraints, can be done.
Okay, thank you very much. But sorry, I have an extra question. I'm just wondering, for example, for one simulations, what's the time cost? If I don't know how much.
For the elastic model?
Yes. How many CPU
the elastic model solves a set of, say, 16 or so coupled differential equations. And it is basically instantaneous for all reasons. So it goes as you watch it. This movie was produced in the time, more or less, that it took you to see it. So it's real time.
Thank you.
Yes.
What about this model? So about this elastic model, so at what energy level, at what this energy density of the thing, this model will fail? Because then you are no longer in this elastic minimum.
That's right. That's right. I should say, even if the functional is elastic, we also include dissipation and interactions with the environment that makes the dynamics nonlinear. But in terms of the energy, it is a harmonic approximation. That is true. So anything egregiously different that happens to the DNA, like it melts locally or changes the structure significantly, that cannot be described with where the fit is of a harmonic potential is not good, is not, you shouldn't apply it there.
So, but can you predict from your simulation, the dynamic simulation, say, can you predict at the energy level, stress level, the model will not be good?
With just the elastic rod model, you can't, because by definition it'll stay elastic.
No, feed your model to atomic simulation.
Yes, in principle. So, yes. So, right. So, in principle, what you could do would be what's called a quasi-harmonic approximation. So, you take your molecular dynamic simulation. You have DNA there, and all the atoms will explore conformation space, and you will calculate the covariance matrix of the atomic fluctuations. So that covariance matrix of the atomic fluctuations corresponds to a multidimensional Gaussian. And that Gaussian has an effective potential that's harmonic. If the quasi-harmonic approximation is not a good approximation from your dynamics, say you're calculating the heat capacity or some other thermodynamic property that's measured experimentally for that DNA and it's simulated with you with quasi-harmonics and it's not good, then you have indications to think, well, this is not really going to work. Even a quasi-harmonic picture is not a good approximation. That's the only thing I can think of in terms of testing. This is a good question. I should think about it more. Yes?
In your experiments, and will the DNA sequence affect your results?
Yes, this is a good question. It is about the role of the sequence of the DNA. For elastic rod modeling, we're studying lengths of DNA that are tens of thousands of base pairs. So there, what is the exact nature of each ACTG is probably less important. So for there, we just use a combination of ATs and CGs that's, let's call it random, and we do simulations on it, hoping that it is representative for a large piece of DNA with a multitude of base pairs. For the local dynamics, in contrast, the sequence is crucial. In fact, the only reason, the most important reason why the CUHKSZ study was prominently featured is not as much because there is this CUHKSZ conformational transition. After all, CUHKSZ base pairs are were seen actually before Watson-Crick base pairs. When CUHKSZ himself crystallized AT and CG without the sugar phosphate backbone, he got CUHKSZ conformation. That's why they're called CUHKSZ, right? And they are Watson-Crick only when the double helix is around. And the reason why the study is important, it shows that CUHKSZ conformations are present in DNA at a sequence-specific. The propensity is sequence-specific. What do I mean? This is an A, yes? And it flips over from Watson-Crick to CUHKSZ. But the probability of it to flip over depends very much on what is on top of it and below it, and perhaps extending even further one base pair up here and there. So the local environment is crucial to the propensity, which actually shows that this could be a second-tier genetic code. Instead of having a codon, three of these bases code for an amino acid, right? Here, what we're saying, if this flips over in a sequence-dependent fashion, there are hotspots along the sequence of DNA that have motions that are different. And when a protein binds here relative to there, the entropic cost of binding, losing this motion, losing more motion here than less motion here, is going to reflect into the free energy of binding of the protein. Okay, to manipulate DNA.
Okay, and do you think the region that has a repetitive sequence will have different mechanic features than the just random?
Absolutely. For example, did you know that there actually exists a crystal structure of naked DNA, AT only, so it's AT, all the A's on one side, all the T's on the other, that is entirely in CUHKSZ? in relatively normal conditions. So, entire AT stretches actually are very prone to Hookestein-based pairing. Also, when you have a CG region that's flanked by an AT region, bases at this boundary also have more propensity for the Hookestein than the majority of the other bases. So it's absolutely important, yes.
Okay. Thank you very much.
My pleasure.
As time strains, this session will end here. If you have more questions, you can always find Professor Ioan to ask your questions. So, an announcement. So, before we go to lunch, all the speakers and attendants of this conference, we should get a picture.